N-glycans because are involved in influenza virus infection. And in the second one, we are trying to characterize these multi-antennary N-glycans since have been described as cancer biomarkers. And in these systems, the standard methods cannot solve the problem of the crowded NMR spectra where we cannot distinguish the arms. Then, some time ago, we decided to try to obtain new parameters with structural information, and we tried to extend the method that was initially developed for proteins, originally by metalloproteins that contain already an ion in the structure. And the idea is to covalently attach a lanthanide binding tag to the molecule, in this case a peptide, and to form a complex with a paramagnetic ion. What happens in the NMR? In the NMR, when you have a paramagnetic ion, signals moves a lot, and this movement is called pseudocontact shift. The pseudocontact shift arises from the dipolar interaction of the ampere electron of the methyl with the nuclei in the vicinity of this methyl. The effect can be visualized with these isosurfaces. This surface is the area in the protein that is affected by the paramagnetic ion. And you can have positive or negative chemical shift changes that are drawn here as red and blue color in the isosurface. That means that the signals can move in one direction or the other way around, and this contains information, because if moves, for instance, in the positive part, the nuclei should be in this area, and if he moves in the negative part, the nuclei should be in this area, and this contains structural information. The most important part to understand here is that the distance dependence decreases with r to the -3. And this, for us, is very good, because just in case that you don't remember, the nuclear Overhauser effect decreases with r to the -6. Then you can obtain only local information. But with the pseudocontact shifts, you can obtain long-range restraints for the calculations. The paramagnetic ion also induces paramagnetic relaxation enhancement effects that can be seen as in the experiments as line broadening, and in some cases the signals disappear, but this is not dramatic since the distance dependence in this case is like in the NOE r to the -6, and then this happens just in the part of the molecule that is very close to the methyl ion. Then we started to try the method, or to test the method with N-glycans. N-glycans are in all your glycoproteins and contains this common pentasaccharide core with 2 GlucNAc units, 1 beta-mannose and 2 alpha-mannose. As you know, further extensions of this core give rise to the different type of N-glycans. For instance, you are very familiar with high mannose because there are a very potent group here working on that. And in the case of the high mannose, these extensions contain also alpha-mannoses. But we are mainly focused now in complex type N-glycans, but our method can also be applied to high mannoses, and we would like to test also with high mannoses. In the case of complex type N-glycans, you have 2 additional GlucNAc units and 2 additional galactoses at the branches. This is the general structure that all of us have in our proteins. In order to test the method, we designed, in collaboration with Professor Carlo Umbersack, this structure, this biantennary N-glycan, bearing a lanthanide binding tag here. And in this spectra, you can see the standard HSQC proton-carbon 13 spectrum of this molecule, and this is like the reference. We always have to measure 2 spectra. One is the reference in diamagnetic conditions. In this case, we use lanthanum that is not paramagnetic. This is the reference. And as you can see here, for instance, for the GLUCNAC units, we have just one peak for both H2 protons. For the galactoses, just one peak for both H4 and so on. We can, in standard experiments, you cannot distinguish the arms because are in quite a similar environment. In contrast, when we measure the same experiment in the presence of dysprosion that is paramagnetic, the same signal splits. Now we have two signals. This is good. We can distinguish all of the signals, not only this that I chosen as an example. We have now one peak for each proton in the carbohydrate. This was our first example, and we were very happy because, as you can see here also, there are one peak that moves more than the other. This one moves more than this. And this is telling us something. This is telling us that the peak that is moving farther is located, that proton is located at closer distance respect to the methyl than the other. Then, in order to understand this, we look in the literature for models of these structures, and we found that, according to molecular dynamics, at least 4 possible conformers have been described for this type of structure. There are 2 families, the extended conformations, characterized for this angle around 180, and folded structures where this B arm is closer to the reducing end as here. Then somehow, just looking at this, we thought that the B arm should be the one that is moving more in the NMR, since are the one that is located closer to the methyl than the A arm. But this was just our first hypothesis. And once that you have the data, you can use this software that is called MSpin and has been developed by the same people as Mestre Nova, that for sure you know. And with this software, if you have a geometry, a coordinates of a conformer, you can back calculate the expected pseudo-contact shifts from that geometry. Then we compare the back calculate and the experimental data, and we see how it looks like. And for instance, for the A arm, we have a very good correlation between the experimental and back calculate data, and more or less all the conformations that are quite similar agrees with experimental data. However, in the case of the B arm, considering just one conformation, we could not fit the experimental data. And as was hypothesized by molecular dynamics, this is very flexible, and we need to consider at least 4 structures to fit experimental data. Then, with this information, by the first time we could obtain the global shape and the dynamic of this oligosaccharide that, when you look from the side, displays this type of transitions, up to down motions, and when we look from the top, displays also these swing motions due to transitions between the different structures. What next? Next, we see if we can study even more complex structures, and now one of my main projects is the understanding of influenza virus infection. Why? Since influenza virus recognize these N-glycans that are quite similar to the ones that I have previously described. The difference is that at the end, you have an additional unit that is a sialic acid. And what is very interesting is that the virus that infect humans, the ones that are dangerous for us, recognize sialic acid attached to galactose in an alpha-2,6 linkage. In contrast, the virus that infects birds, the avian virus, recognize sialic acid attached to galactose in an alpha-2,3 linkage. It's a small difference, but it's very dangerous, since when these viruses that are usually in birds mutate one of their proteins and are able to recognize our epitope, this virus is very dangerous because it's the one that spreads very fast between people because we don't have antibodies against this virus, and is the ones that are pandemic, as the last one that happens in 2009, and a lot of people died. Then this is the scheme of the virus. There are two proteins at the surface of the virus. One is neuraminidase, that is involved in viral release after infection, and there is another one that is called hemagglutin, that is the one that we are studying, that is involved in the recognition of this sialic acid. And this molecular recognition event is the first step in the infection. Sorry. Then we are trying to understand how this molecular recognition event happens. We are collaborating with Professor James Paulson group at the Scripps Research Institute in US because his group published this paper where they found by using glycoarrays that the new viruses that are evolving, for instance, this H3N2 or the one that produced the pandemic infection in 2009, not only like the sialic acid at the end of the arm, they prefer also long chain structure in the recognition. And the group of Paulson's group is trying to understand why this evolution of specificity goes to these long chain glycans. This is a hypothesis. This is a model, computing model that has been calculated in Professor Robuff's group, and they postulate that these long chains can simultaneously bind to two carbohydrate binding sites in the hemagglutin trimer. Hemagglutin is a trimer with 3 binding sites for the sialic acid, and the idea is that perhaps these long chains can simultaneously bind to 2 of them. But this is just a model because they have crystallized the protein, but there are not enough electron density to describe the carbohydrate, as is usually the problem in protein carbohydrate complexes. Then, in this context, we decided to try to see if we can say something by NMR. But in order to distinguish the arms, we need the lanthanide, and we could synthesize these structures with the lanthanide binding tag. This work is the thesis of Beatriz Fernandez that is working with me, and she has studied these structures. In this case, we have removed the first GluCNAc of the core of the N-glycan in order to have the lanthanide binding tag closer to the end of the arms, because these ones are bigger than the previous one. And we have already studied the compound with just 1 LacNAc repeat and with 2 LacNAc repeats, because the idea, according to glycoarrays, is that the compounds with several LacNAc repeats have higher avidity for the protein. Then I'm not going to show you all the spectra, I promise, but I'm just going to show you this because for me it's beautiful. In this case, this is the compound, the B antenna with 2 LacNAc repeats. These circles, yellow circles, are the galactoses, and as you can see here, we have 4. In the standard experiments, you have just 2 peaks. In this orange peak, you have 2 galactoses, and in this other orange peak, you have the other 2. But when we acquired the experiment with dysprosion, we have 4 peaks. And this is nice because we can again distinguish every this is just an example because it's one of the parts that I really like, but we can also distinguish the sialic acids and most of the signals of the molecule, and this is a very long chain. And once that we could assign the experiments, that is not trivial, is something that is not that simple. We could assign all the signals, combining different NMR experiments, and we could derive the conformation. And it's nice because if you look here, this is the conformations for the shorter chain, and here you can see the conformations for the longer chain. And as you can see here, the LacNAc structure display both extended and folded structures. We can see that this chain, this is the overlay of different conformers, is moving a lot. In contrast, the smaller glycan is less flexible. Most of the conformers are in this area. And this makes sense because if you look here, the sialic acid attached to galactose in this alpha to 6 linkage is like a hairpin. And this hairpin cannot be accommodated in folded structures in this small chain because collapse with the main chain of the A arm. You need the additional units to have the hairpin located at longer distances respect to the core. And this can be important in the recognition of the protein, since in order to reach two different binding sites, you need to have this flexibility to accommodate. This is just a hypothesis, but we think that is important, the flexibility in the binding in this case. And what next? Next, we are going to study the real problem. Now that we can derive the conformation and we can distinguish the arms, we are performing binding studies with hemagglutinins. The hemagglutin is quite difficult to obtain. It is expressed in mammalian cells, and this has been done in San Diego. Beatriz spent some time there during the thesis in order to learn and in order to express the protein, because carbohydrates are quite difficult. And not only this protein recognizes carbohydrates, but it's also glycosylated itself. And the glycosylation of hemagglutinins is important. Then you cannot obtain a recombinant form of hemagglutinins in bacteria, for instance, because then it's not glycosylated. Then you need to express the protein in human cells, but they have developed the protocol. And now we have something that is very nice because we have different variants of the viral protein. For instance, here I'm going to show you the comparison of the Hong Kong 68 hemagglutin. Usually, if you are not familiar, the name of the virus is given by the place where happens the first infection. Then this one happens in Hong Kong in this year, in 68, and this one is very new virus that the first time appears in Victoria, in a city, in 2011. Then we choose these two to start with. Why? Since according to glycoarrays, this Hong Kong 68 variant recognizes mainly sialic acid, but the length of the chain is not important, according to glycoarrays. But in this case, they detect, is the case that I saw before here, they detect a strong influence of the length of the chains for this Victoria 11 structure. Okay, the model is for Victoria 11. Then we study the interaction by using saturation transfer difference experiment that you are using also a lot here. And we found something that is very nice, and this is just the off resonance, that is the reference, and this is the STD. It's the thing that you have to look. And if you compare the STD of the same compound in one case and in the other, you can see that are different. For instance, these two peaks are very small in this, and these are much higher in this. And what means that? If you have a peak in the STD, you have an interaction of that protein. That proton is closer to the protein, and in this case, the protons that are closer to the hemagglutin are mainly the protons of the neuraminic acids. Neuraminic is the same as sialic acid. The end of the chain is important for Hong Kong 68, but the other GLUCNACs that are inside the chain are not so important. The STD is very, very small. In contrast, in Victoria 11, we have a strong STD for the sialic acid that is expected, but we also have a strong STD for the internal units of the chain. And what does this mean? This means that there is an extended epitope. Not only the sialic acid is important, but also the other units of the chain are closer to the protein surface. And this is in agreement with the glycoarrays and is giving a lot of information. We are trying to go even a step further. This is the Tri-LacNAc structure that is really huge. The postdoc that was working in San Diego doing this moves to China, say no, forget about that. I'm not going to continue because this is not doing by traditional organic synthesis, it's doing enzymatically. In San Diego they have set up every glycosidation step with a proper enzyme that is able to do this, but this is a lot of work, is not easy. This structure is quite challenging. Then today we only have access, to the compound without the lanthanide binding tag, but we are trying to convince Wenji to do this. He's doing now in China. He has started his own laboratory because he is really, really good in synthesis. And now we are trying the coupling. He promised to me that we are going to have the molecule soon. But at least he sent me this. That is a lot. It's really impressive. This is the longer carbohydrate that we have studied ever. Is quite huge. And in this case, as we don't have the lanthanide, we cannot distinguish the arms. But anyway, we have run the STDs because we can obtain information. And it's very nice to see that we obtain a similar behavior like with the LacNAc structure. This is the STD with Victoria 11. This is the STD with Hong Kong 68. And as you can see here, for instance, we have always the STD for the neuraminic acid, the sialic acid, but again, it's the same in this case. For the GLUCNACs that are inside the chain, we only obtain high STD effect in the case of Victoria 11. It's like Hong Kong 68 just likes the sialic acid. The rest is more or less the same, but the other is more specific. Then this is the model made by Rob Boots that kindly provide us the coordinates. And once you have a model, you can back calculate with this program the expected STD. And we have done this with the model, and the predicted STD values are in good agreement with our real STDs. Then this is not an X-ray structure, but at least now we can validate that the model fits some experimental data, and we think that perhaps is the case, as is postulated, that these long chains can interact with a huge surface in the hemagglutin. Once that we could study this structure with the lanthanide, we could also distinguish these units, and this is important since if this is the case, we are going to see also STDs for these units that we cannot distinguish now with the information that we have, but we are working on this because we think that it's a hot topic now in the area, because it's like the Game of Thrones, winter is coming, everybody says that the new pandemic is coming, and we have to understand as much as possible about this in order to design inhibitors. There is a very nice work done in Utrecht by Roland Peters that he has designed a dendrimer that is multivalent with this information into account, he has placed 2 inhibitors of these structures at the proper distance, and it works. It works. Then it seems that if this is the case, it can be very important to understand this type of structures to develop proper inhibitors, because there are no inhibitor described for hemagglutin. I don't know if you remember this famous Tamiflu that was developed after the pandemics, but Tamiflu is for neuraminidase, not for hemagglutin. Then any advance in the development of inhibitors of this structure could be very, very important. And now I'm going to move to the second line. I hope you don't look like this now, a bit at the end, but this is important because we are very complex. We not only have biantennary structures, we have triantennary, tetraantennary, and even more complex structures. Then we need to develop tools to study this, because there are information on these structures that we cannot study if not. And also this biantennary, monantennary, or whatever interacts with things, like in these pictures. Then you need to develop tools, or at least we are trying to develop tools to study this. Then again, thanks to the great Carlo Umbersack, that spent some time in Seville, I think. He likes a lot Seville. He could synthesize this tetraantennary structure, and this is regular chemical organic synthesis of carbohydrates. Imagine, it's wow. There was the thesis of one student to synthesize this molecule, and with the lanthanide binding tag. Then we have, in this case, 4 arms that cannot be distinguished in the standard experiments. This is just an example because I really like this example. This is the standard proton NMR of this compound with lanthanone. This is the aromatic linker, but the carbohydrates are mainly here, the carbohydrate signals. This is the same with dysprosion. Wow, looks like a protein. For me, it was like, I also have been working in proteins when I was a child with Pedro. Do you remember that? I was always complaining, no, proteins are too difficult. He said, no, be patient. But are very difficult, I have to say. But for me, this when I looked this, it was a carbohydrate that looks like a protein with a lot of dispersion, and for me, it's beautiful. I'm a bit freaky about NMR, but it's very nice experiment. Then, and what is even nicer is that when we repeat the things with dysprosion in the HSQC, we have 4 peaks. For the galactoses that are overlapping, just 1 peak in black for the 4. In the presence of dysprosion, we have 1 for each one. It's like, oh, it's not that difficult. This is nice. We can see all the signals resolve. This was very important result in the field, because it was first time. Then the conformation of these things are quite difficult to study. Now we have not only 1 alpha 1,6 linkage, we have 2. Then this is a very flexible thing in principle, and we should consider several conformers in order to build models. But at the end, what we have realized is that we could, this is the AB and this blue is the C arm, we could fit very nicely these pseudo-contact shifts, considering just 1 conformer in the middle linkage alpha to 6, and it's the Gauge conformation. But what is important here is that somehow the presence of the additional arms restricts the flexibility of the molecules, because now not every conformer is possible because collapse with other parts of the molecule. Then in principle, it's not that flexible. At least the core of the N-glycan with all the arms is more rigid than the biantennary structure. And thanks to Armando Vázquez, that is now in Brazil, and is the scientist that has developed the program, he helps me a lot in order to back calculate the pseudo-contact shifts of this structure, and we could derive that mainly displays this type of movements of the D arm. The core is quite rigid, but the D arm, the additional beta-1,6 arm, is flexible. And this is important, since there are a lot of papers in the literature that say that when you have this additional arm in your glycoproteins, is not a good news, because it's related to cancer. There are like a type of we are trying to understand the glycocode, what means every single change in the structures, and there are some of them that are not a good news, because when cells start to behave in a strange way and produce also glycosylation patterns that are different to the standard ones. And this, in the standard structures, you usually don't have this additional beta-1,6 branch, and is a lot of things have been done yet. This is just starting, but it seems that it's related to several tumors. Then in this case, we have also performed binding experiments to see if we can derive information about the recognition of these structures. And again, we have used STDs because are very powerful here, that we can distinguish the arms. And for instance, with Ricinus communis agglutinin, that is a protein lectin from plants that recognize galactoses, we have similar STDs for all the arms. Then this lectin can bind to all of them. But in the case of Datura stramonium lectin, that is described, that prefers the six arm, the arm, we obtain all higher STD effect for this arm. Then we could fit the biochemistry data that has been postulated for a long time, but there were no evidence, because there are not X-ray structures of these things or NMR studies in detail where you can really see what's going on. Then what next? This is what we are doing now. This is the thesis project of Monica Martinez that is also working with me, and the idea is that now that we have set up the method for these tetraantennary structures, we are going to study in a systematic way the different modifications that we have in the proteins to try to understand what is doing this in the structures. For instance, there are some cases in where you have an additional bisecting unit at the core. There are in some cases where you have a fucus at the core, and this is also not a good news because it's also related to cancer. There are a lot of people now working on core fucosylation of N-glycans because it's especially overexpressed in tumoral cells. And we are also studying the compound with both modifications of the fucus at the core and the bisecting unit. I'm not going to show you all the spectra, but this is very nice. This is the first experiment that we measure with this bisecting GLUCNAC. And it's nice because you have 1, 2, 3, 4, 5 acetyl groups of the GLUCNACs, and we have all the peaks in the methyl group. And this is not the case in the standard experiment that you have like a mountain, a big one with all of them. But now it's like we have everything. And for instance, the bisecting unit is quite different to the others. Then for doing interaction studies, this is very, very important. And something that we have discovered, and this was also amazing for us, is that we try to measure the standard NOE of this, the NOSI of these structures, and thanks to the good resolution that we have for the methyl groups, we have methyl groups that are in regions that are almost alone, we could detect an NOE. You can say, it's not very nice, but it's nice because it's not that easy to measure this. We could detect an NOE in the bisecting structure between one of the arms and the GluCNAc, this one in blue, at the core. And that's only happen if you have a folded structure, because this NOE cannot be explained with the standard structures, because in the standard structures, this is far apart from this one. You need like to fall in order to reach this GluCNAc that is at the core. And then this is amazing. Just one additional GluCNAc in a structure matters, since in the compound without the bisecting unit, we don't have the NOE, and we don't have a high, there is nothing here, no NOE, very, very, very weak peak. Then the conformation is mainly extended in the compounds without the bisecting unit. But when you have the bisecting unit here, this makes like a switch, and this folds, and then you can detect the folded structure in the compound. Why we think that this is important? Because there are several crystal structures of bisecting N-glycans with lectins where the bound conformation is folded. And then this is not that this happens just in the bound state. It happens also in the free state. Somehow the molecules are pre-organized for recognition, and this is the first time that this can be seen thanks to the good resolution of the experiments. This is not published yet, but we think that is a very nice result. We are also comparing the structures with the fucus. In contrast, the fucus is like, we are thinking on that because the fucus, in principle, doesn't affect too much to the global shape of the molecule. It should be a different level of regulation. The fucus should be important in the interaction with other things, but the conformation of the sugar is the same with and without the fucus. We have studied other fucus derivatives, and is more or less, the behavior is more or less the same. Then I think that there are different levels of complexity in carbohydrates. In some cases, an additional unit modulates the conformation. In other cases, modulates the interaction, are different mechanisms. For instance, in the compound with the two things, with the bisecting unit and the fucus, we have again the NOE, and we have the folded structure. Then in each structure with bisecting unit, the folded conformer is relevant. Without, in principle, you can have extended and folded conformations. We are also studying the interaction of these structures, in this case with galectins that recognize beta-galactosides, and we have studied galectin-3 and galectin-4. But in this case, we are not seeing clear selectivity. Galectins are a bit promiscuous, I have to say, and they bind equally more or less to all the arms. And this is an ongoing project, but in this case, we could not see any selectivity. All are involved. The idea, there is a recent paper also of Carl Umbersack with Rob Boots that postulates that in this case another protein can bind to this part, and this is like a sandwich, and could be the case because we also have STD for the C-arm. We have for all of them. It's like perhaps in this case it's like a cluster effect, and you can recognize as much galectins as you can. And in this case you have STDs for all of the units, but we are working on this. Okay, and just to finish as an overview, we are usually draw the carbohydrates like this, but are not like this, are like this. And like the take-home message is that you have to start thinking in sugars like this. Because when we try to figure out the interactions, you cannot think in something that is like pointing to that place, because it's not like this. It's more or less like this. And I like a lot this type of representation where you can see all the space that is available according to excursions of the molecule, because this gives a lot of information about the possible interactions and the binding mode. And our idea is to continue with this, trying to figure out both the shape of these complex things and their interactions with other molecules. And this is all more or less. I'm going to thank my ladies. Beatriz, that is going to defend the thesis next year. Monica, that is in the middle, more or less. Javier Rodriguez, that was a postdoc that is not currently working with me, but he helped me a lot in setting up the molecular dynamics in order to study these complex carbohydrates. And Javier Cañada, that is always collaborating with me from CSIC in Madrid, in CIB. Jesus Jiménez Barbero, that is now in SIC BioGune. And all the foreign collaborators. I'm trying to start my own research group, then I only can do this type of so difficult things thanks to these people in Universidad San Pablo CEU, where they are designing and synthesizing the chelating units, in Scripps Research Institute, where they are working in these long chain structures and the hemagglutinins. In Germany, Carlo Umbersack is doing this type of multi-antenna presentations. This is the NMR facility of my university. We have a 700 with cryoprobe and another smaller magnets. I have seen your beautiful 600 that you have here. That is also very nice. But if you want to move from the hot in the summer, you can also come to Madrid if you want to measure in our 700. We are always open to collaborations. And I also would like to thank the funding. I applied yesterday for a new project. I hope, please cross all your fingers for me, because it's difficult to start the group, but okay, I'm lucky because I have a project in my previous application, and now we will see if I can continue doing these things. And thanks for your attention.
Many thanks, Ángeles, for a wonderful talk. I think we have to keep with the microphone for our 200 followers today. And so, questions, comments? Okay, Pedro.
Ángeles, very nice talk. Really, very nice.
Thank you.
One thing, have you realized that you have measured a long-range NOE in a sugar?
Yes.
Yeah? Because this is not common in sugars. In fact, it's quite rare.
Yes, we are very, very, very happy with that, and now we have like changed our mind, because we always say, no, this is too difficult, we are not going to measure the NOE. But now we say, wow, we are going to measure the NOE in all the samples, because sometimes I'm going to say something in Spanish, sorry, porque mi abuelo, que era andaluz también, por eso a mí me gusta mucho Andalucía, decía, antes de echar a correr, mira a ver si puedes andar. And remember, to my grandfather, this is the case. Sometimes we are trying to develop quite complex NMR experiments, but we forgot that the standard ones are also important and work. And in this case, it was for me like, wow, I'm trying to measure pseudo-contact shifts at 40 angstroms, but the NOE, that is the standard one, it helps a lot in this case. Because it was clear, if you have the NOE, you have the NOE. Then these are close. And the PCS, sometimes we are trying to do a lot of fittings, and sometimes it's easy, and other ones you are not so clear if it works. But when you have both, it's great, because you define the structure. Then we are like changing the protocol now. And I say to Monica, measure the NOI always, just in case. And we are also measuring in water, because the NHs of the GLUCNACs can be very nicely seen in water. And then we are also measuring the NOIs in water, in D2O. We are like changing the protocol, because sometimes you discover things that you don't expect with the standard things that you should have tried at the beginning instead of very difficult things.
Right. Thanks. Another small question. In the project of the agglutinin, sorry, no.
Sí, hemagglutin.
Hemagglutinin, yes. You have two different strengths, and the STD have different intensities. Do you have an idea about the affinity of the different compounds for the different proteins?
No, and this is something that we are trying to do, because they cannot do it either. And then they ask us, and what we are going to do in the next project is that we are going to try to do competition experiments with this method that is something very new. Because if you can label one of, for instance, one of the compounds and the other no, you can distinguish, for instance, linear, monoantenna and biantenna, one label, another one non-label. Then we can perform competition experiments and see if the binding affinity is the same or not. We have already tried with the non-label ones, but the spectra is the same. With one chain or two is the same. Then if you cannot distinguish the signals, you cannot really see what is going on. But this is something that we are trying to do, because it could be like distinguish the same thing in different presentations. Then you can perform relative binding studies and things like this.
Right, thank you.
Welcome.
Well, thank you very much for this excellent lecture. It was really impressive. And I think that we don't have to cross too much our finger for you to have the next project. I'm pretty sure.
You never know. Just in case, do for me, please.
Well, I'm not very specialist in the field, but the first question is why this tag? Why this chemical tag? It's because it's the only one. It's because of its rigid. It's better if it was flexible.
No, this is a good question. Is because if rigid, because we have some bad experience trying to do, because for instance, is commercial a precursor for doing this that has an asparagine unit here, because in glycoproteins you have an asparagine. Then is in principle is normal to use something like an asparagine here. But we try to attach the linker to the asparagine, and it's not working, because if the movement is too much, then the average of the effects are killing the pseudo-contact shift. You have pseudo-contact shift, but it's very, very small. You need something rigid. We are trying to improve, because we have a problem that our design that has been developed, but there are very few groups working on this. Just a Japanese group, that is the one that published these high manos with the chelating unit, Kato groups. And they are also working with these units, but this is not symmetric. And this is a problem, because you can have movement, and the movement displays the methyl in a slightly different position respect to the carbohydrate, and you have to consider also that in the calculation, and it's not very nice. Now we are moving to other linkers that are symmetric. That is better for us. Rigid and symmetric is better. and also with more coordination points, because we would like to, for instance, I know that you are interested in this design and this type of proteins, but in that lectins you have a methyl in the binding site, and that methyl could exchange with the lanthanide, and we don't like that, because in that case you don't know what are you seeing. Then for moving to C-type lectins, we need to have more coordination points here, to have like a cage, to have block the methyl here, and it's not that easy. We are trying the synthesis, but it's not that easy. Now we are trying to synthesize one with 8 coordination points in order to move to other lectins. But these things are described, but are not that trivial to do.
And the next question is quite philosophical. You have shown the importance of carbohydrate and in these flu things, but the end it's so complex that once we know, we have the information, what we can do with this information to go further, to develop something useful.
For instance, this work that I mentioned about this Roland Peters is like a clear example that this is important to note for the design, because if he says if the distance between this carbohydrate binding site and this other is 40 angstroms, I'm going to design a synthetic molecule with that spacer, with one inhibitor in one place, another in the other, and see it works, and it works. Then sometimes the rational design works, others no. You have to do both, but in this case, at least the first inhibitors that has been tried based on the information available obtained by extractors is working. What next? If you have a pandemic with one of these viruses, you cannot design an inhibitor with just a sialic acid mimic, because the other parts are also very important. Then you need something with bigger. If not, you cannot compete with the glycans. If you have a strain that just likes the end of the arm, then it could be easier to target that. But I think that to know this is important, to be protected, because if not...
I don't mean that it's not important. I mean it's so complex.
Yes.
Yeah, that at the end, and you say that the synthesis of complex carbohydrate is not solved, even though there are some people saying that you can do that automatic synthesis of carbohydrate. And I mean, yes, the next step is to simplify, but can we simplify at the point that we can have very small molecule or economically useful?
I think that for some variants, yes, for others, no. But I think that a small molecule is not going to be enough to interfere with such a extended epitope. For me, at least with this mechanism, you can think in other things. But if you want to target hemagglutin for some of these variants, you need to have something very powerful to interfere with this network of contacts with the surface. And perhaps this is the reason why there are no drug in the market, because it's not that small. If you disrupt one interaction, is enough. No. It's like very difficult, yes.
Yeah. Well, thank you very much. It was very nice.
Welcome.
Very nice, thank you. I have a question regarding the conformational analysis of your oligosaccharides. Okay, you show that in some cases one of the arms come in fact close to the tag, so
yes.
to the let's say reducing end of the oligosaccharide. But in the real situation, there is not the tag. There is a protein. This is an N-glycoprotein, which
yes.
is very big. So
yes.
my question is: the presence of this protein there in the real situation would condition the conformational space available for the oligosaccharide, and how do these influence the conformation of the oligosaccharide in the real settings?
Yeah, this is a good question, and this is my next project. We are going to try to implement this in the context of the glycoprotein. because it's very, very difficult, but it's the case. Sometimes it influence a lot the protein and others no. I have been studying a lot the literature to write the grant, and there are very few examples described, but for instance, in one very nice paper that from the group of Prestegar, they studying an antibody that is glycosylated. In that case, one of the arms interact with the antibody, and then the conformation is modulated by that interaction. But there are also a recent paper from Jesus' group, I'm not involved, but has been done the last year of Luca Unione, where they study other glycoprotein, and in this case, there are not a big influence. The carbohydrate is far apart from the surface of the protein, and there are not interactions with the chain, but there are not systematic studies because X-ray crystallographers don't like to study glycosylation forms because they cannot obtain a crystal because are very heterogeneous, and then they use systems to express proteins without sugars. And then I'm trying to ask people, are you working with, no, I always select a cell that doesn't glycosylate in order to have an X-ray structure. Then I'm going to try to express a glycoprotein in human cells, because we have been discussing previously is the only system that produce a glycosylation similar to our, and I will tell you. But there are no tools yet, and there are not information, because in the PDB there are few structures, and most of them are of the same protein. There are not the diversities, that there is only one that is a fragment of an antibody that is very stable and you can produce a lot, and then everybody studied the same protein. Then it's not enough information to say that the protein matters for sure. Always, we don't know. We are going to try to study this.
Thank you. Next time.
Next time. I hope it works.
Next.
Thank you, Ángeles, for the nice talk. So my question is regarding the tetraantennary and the interaction with galectin. So you say that probably one arm is available to interact with a second protein.
Yes.
So how you try to perform isothermal titration calorimetry studies just to see if you are able to see complex 2-to-1?
No, but we would like, and we can do it because this is ongoing and we have samples. Then we can do it. It's a good question. Because at the beginning we have had a lot of problems setting the protocol, but now that we have the information, we can try to do additional things. Yes. This is very new. It's nothing is published about this part, and we are working. Then it could be nice to measure other things to see the stereochemistry, no, the stoichiometry of. It could be. And
a curiosity, just in the same case. So you located basically all the acetate groups, except of the N-acetyl glucosamine that's closer to the lanthanide.
Yes.
Where is this?
Is missing because this relaxation enhancement effect that I mentioned
so it's too close.
Is too close.
Okay.
Yes, this is the only one that we lost due to the relaxation enhancement effect. We don't know where it is. Could be. Yes, in Madrid perhaps. Yes, no, it's amazing because some things move a lot, move really a lot. The aromatics are also missing, because are very close to the paramagnetic ion. And this is also something that we don't have explored, but you can explore, because for instance, if this process is quite strong, but if you put gadolinium here, that is the one that is used in imaging, then you can explore this effect, because gadolinium is like a hole in the spectrum. Everything that is in the environment disappear. Then imagine that you study the interaction with something with gadolinium here. Even if it's weak, you are going to see the effect because it's huge. And then there are some people that for doing screening, even in the pharmaceutical companies, are doing this. To see with binders, they put something very strong like gadolinium, and if something is close, the signal decreases. And then you see that it's close to the other part. This is not such elegant because we like to see its proton, what happens if it moves or not. But if you just want to see yes or not, recognize something or not, you can use gadolinium, and then the signals of everything that is close, just.
So for this kind of
Decrease.
experiments, this Procion is the best one.
For us, till this moment, yes, because we are seeing effects till 40 angstroms. That is quite far. For us, is the best till now. But if you want to study a disaccharide, no. Then you can use thulium or europium or other one. But for these long things, yes, this Procion works quite nice.
Okay, thank you.
Anyone else? Nope. Okay, my quick question. It's amazing what you show us about the NMR, what you can do with the conformations of these molecules. But I was just wondering how difficult it is to make. Well, how long does it take to make one of these huge carbohydrates, and how do you make them? You put one monosaccharide each time, or you make the arms and you stick it to the antenna at the end? And at the end of the day, how long does it take? How many steps and yields?
And for instance, Beatriz, my student, spent three months synthesizing just one linear one. For instance, Beatriz synthesized the Tri-LacNAc structure, but not the B antenna, the mono antenna, in 3 months. But she learned everything from the beginning. Then perhaps if you are used to do that, you can do it in 1 month. But there are a lot of steps. And in this case, the purifications are easier because as is an enzyme and works in a perfect yield, you don't have other things. You have just your compound, and you have to remove the initial material, and that's all. But you don't have mixtures of things. Then the purifications are much easier with the enzymes. But for the multi-antenna, this is very, very long. The student that is doing this is now writing the thesis, and I asked him if he could send me more amount of one of these ones, and he said, forget about that. No, because this is done with, these ones are done with a standard organic synthesis, and they use just enzymes in one step. And the others is the ones that you are doing, and you know better than me, and it's very, very hard work.
I can imagine.
Yes.
I cannot imagine. Okay. Is there any other question? No,
Just one short comment. What I just want to say is that I'm doing most of these things with 1 or 2 milligrams.
Wow.
So all the project. Then it's very difficult, but at the end I don't ask for more than this, because if not, they are going to say no. But with 2 milligrams, with the cryoprobes that you have here, you can do it. You can use the same sample for the characterization and for the binding studies. You have to be very careful with everything, but I don't use more than this, never. With 2 milligrams, I can do a lot of things. This is the good news.
Okay. Thank you. So if there is no other question, let's thanks again, Ángeles, for a wonderful talk.
Thank you. Bien, yo creo que bien, ¿no?